Swayam Prabha. Digital India. Educated India.
In this lecture, we will discuss about the applications of the bioinformatics tools or the algorithms to structure prediction and folding rates and folding stability and so on. So, in this class I will mainly focus on predicting the 3D structures of proteins from amino acid sequence, right, using the bioinformatics tools. In the previous class we discussed about various parameters or the properties which can be derived from protein 3-dimensional structures. Right. What are the various parameters we discussed in the earlier classes? Contact maps, solvent accessibility, buriedness, solvent accessibility reduction ratio, transfer free energy, and based on the contacts you can see the contact order, long range order, multiple contact index. Then other parameters like as hydrophobicity and the interaction between the two different residues. What are the potential interactions, right, between two different residues based on the type of residues, like the hydrophobic interactions or cat and pie interactions or electrostatic interactions and so on. And later on we discussed about applications of the contact maps to superimpose protein structures. If there are two different structures, you can see how far they are similar in 3D structures, right. We discussed about the superimposition of protein structures based on contact maps. So, when we look into the availability of 3D structures in protein data bank, at the moment we have around 133,000 structures in protein data bank. On the other hand, the available information regarding protein sequences in which database? UniProt database, right. So, we look at it is currently about 89 million sequences, right. So, if you compare the availability of amino acid sequences and structures, right, available sequences are about 700 fold than the availability of the structures, and the protein structures will be helpful on understanding the function, identifying the active sites and the antigenic sites, right, or the different binding sites with other complexes and so on. So, it is very important to have the structures of proteins, right, than sequence information. Due to the availability of more number of sequences and less number of structures, it is important to predict the structures of proteins from just amino acid sequence information. So, we look into this structure determination. So, what are the various methods used to determine 3D structures of proteins? X-ray crystallography, NMR spectroscopy, right. Even with the latest information as well as the refined instruments, right, and well sophisticated apparatus. So, you can see the structures, available structures are only 133,000, and to determine the structures of a single protein, right, it may take about $10,000 $15,000 and at least 2 months to 1 year if the structures are simple. When you go with the complex structures like protein DNA complexes or protein protein complexes, right, in this case you will get more time, for example 3 to 4 years, right, to understand the structure and function, right, as well as the expense is also very high. So, comparing the time to determine the structures of the proteins and the complexes, right, as well as the cost of getting the structures, it is very important to predict the 3D structures of proteins from sequence. So, deciphering the native conformation of the protein from this amino acid sequence, right, this is called protein folding problem. In 1963, Anfinsen stated that the amino acid sequence contains the information regarding the structures, and in this case it may be possible to get the 3D structures from just amino acid sequence. So, what is the protein folding problem? It is getting the 3D structures of a protein, right, from just its amino acid sequence. How to get that? If we took the 3D structures, first you can see this elongated chain, then it will distorted here and there. After that you can see the probable bends, right, and finally it will get the regular shape, right, and kind of 3D structures, right. This is the animation. You can see that how from the unfolded state of a protein finally folds into the stable 3-dimensional structure. So, how to obtain this structure from amino acid sequence? There are various methods to predict the 3D structures just from this amino acid sequence. One of the most popular methods, right, that is called homology modeling. It is based on the principle that if two sequences share high sequence identity or sequence similarity, then the assumption is that the structures are also similar. Based on that principle, so they developed the methodology called the homology modeling or comparative modeling. If there are no significant homologies available, then the another method is whether you can recognize the folds, whether they can make any specific folds, and if that also fails, then you can try from the beginning. That is called ab initio method, right. You can start from the scratch, starting from the single atom and build the second atom based on the bond length, and the third atom based on the bond length and bond angle, right, and the fourth one with the bond length, bond angle, torsion angles, right. You can build up a method that is called ab initio method. For example, this atom number 1, you can fix the second atom, right, this one, this 2, right, just with this bond length. If you know this atom is C and this is also C, you can know the bond length of C and CC. So, you can fix this second position of the second atom. Then for the third one, for example, if you put it here, so what determines the position of this third atom? Angle. One is this length, and second is the angle, right, this theta and length. So, this will determine the location of this third atom. So, here the fourth one, right. So, here we require 3 parameters, one is the length, and here we need the angle, and then we need torsion angle, right, takes different conformations. Then we go with the fifth one, we have 3 information, you can 3 measures like length, angle and torsion angle. With respect to these 2, 3, 4, you can fix the fifth atom. Likewise, we can grow and finally make the energy minimization to get the final stable structures. I will discuss about these details in later. Then recently there are several methods called hybrid method, right. This combines different techniques. Wherever we have the significant similarity or the homology, so we can take the structures using homology modeling, and if the structures are not available or the sequence identity is less, then we do the ab initio modeling, and finally we combine everything together and make a single structure using energy minimization. So, this model also works fine, right, recently these hybrid models. The ability of these techniques can be assessed using a competition called the critical assessment of protein structure prediction of proteins, it is called CASP. This is conducted once in 2 years, right. We can enter into the competition, and we can also try to build our models and see how far our method can be accurate, right, compared with the other methods in the literature. So, let us start with the prediction technique. So, if you have the protein sequence, I will get the experimental data, you can get the protein sequence. So, we want to predict the structure. First, see the database searching, right, available database you can search. Which database you need to search? PDB. You can say PDB, you can use BLAST, use the database, the PDB database with known structures, right. So, if you have the significant homology, significant identity with any of the structures, you can make the multiple sequence alignment, right, and see the conserved regions, and if you find significantly homologous structures in the PDB, yes, then you can go with the homology modeling. So, which type of identity is good to hold the homology modeling, that I will explain very soon. So, if you find significant homology, then you can go with the comparative modeling or homology modeling. Fine. If there is no homologous in the PDB, right, if it is no, then we go with the next step. You can try to do with the secondary structure prediction. If you have the secondary structures, then based on the secondary structures you can try to predict the fold, which type of fold it can make, right. If we can predict any specific fold, then we can align with these structures, and then based on that information you can go with this homology modeling at the particular alignment with the particular positions. Even that fails, then we go with the ab initio modeling, start from the scratch and then try to get the 3D structures. The performance and the accuracy depends on various factors. For example, if you have highly homologous structures, then you can try to achieve the highest accuracy, right, close to the experimental values. Less homologous, you can do the ab initio technique, that also you can predict with reasonably good accuracy. So, let us discuss about the homology modeling. What is the principle used in homology modeling?
Similar
right? You predict the 3D structure of your protein, right, from the sequence with an accuracy which is comparable to the experimental data if the identity between these proteins are too high. For example, here I give two different proteins, here the identity is 84% with 92% similarities, and if you get highly homologous sequences, you can get a model which is similar to the template what you choose. So, you can assume that the two sequences they share very common residues, have high sequence identity or sequence similarity, then these two proteins have similar structures. So, if you see these structures, they can show in the two different colors, one is in blue and one is in grey. So, they are also aligned well in the sequence, you can see the similar structures with the less RMSD, that is about 1 angstrom. So, this is a principle used in homology modeling. There are also instances in which the sequence identity may be less, but they can have similar structures. For example, the temporal fold, these proteins they share very less sequence identity like less than 30%, but they have similar structures, right, with the high similar structures you can get in the case of temporal folds. So, how this protein homology modeling works and why we need to model the protein using homology modeling? Because if you want to do structure based drug design or to understand the important functionally important information for a protein, it is good to generate models using in silico protein structure prediction. So, here I list up some of the important applications. One is for any target, if the structure is not known, to identify the small molecules which can potentially interact with these targets, we require the structures. If the structure is not available, then it is very important to have a structure, right. In this case, if you generate a model, then this model could be used as a potential target for identifying the lead compounds for any drugs. In the structure based drug design, it is required to have a model and also to find the protein functions, which residues are important for the several protein functions, right. You require the structures, then also you can see the antigenic behavior and the rational design of proteins with increased stability or functions and so on. For all these aspects, modeling is the only way in which we get the structural information, right, in if the experimental techniques fail to get the structures, right. To use a X-ray crystallography, which is required for X-ray crystallography? Crystal, right. If your protein is not able to crystallize, then you cannot use X-ray crystallography for determining the structures, right. And if you get big proteins, right, sometimes you cannot use NMR spectroscopy. If the experimental techniques fail, then the only option is modeling. In this case, we have to use the modeling techniques for predicting the structure. For example, membrane proteins, it is very difficult to crystallize, right. In this case, you can use these modeling techniques to get these 3D structures. So, how we get the conclusion that you can get the structure from the sequence? Because if you have the sequence, the residues are accommodated in different specific range, right. Also, you can see the specific combination of the amino acid residues. Then we discussed about the different parameters, structural parameters, right. You can see even they are specific combination. Some residues which are far away in the sequence, they are also close in space. Essentially, if you see that the sequence contains the information regarding the structure. For example, there are several hydrophobic residues. These hydrophobic residues tend to form this core in the 3D structures. And if you have one positive charge residue and negative charge residues which are very close in the sequence, they have the tendency to form the ion pairs, right, or solvents, right. So, you can see that the sequence contains the information regarding the structure and it is possible, right, to predict the structure from its amino acid sequence. So, now the question is, if two proteins have different sequence identity, for example 80% or 60% or 50%, how accurate you can obtain the model? Here I can see that if it is more than 60%, in this case you can get a model which is similar to the medium resolution NMR structures, and you can also the low resolution crystallography structures, you can get up to that level, right, that is very high accurate, you can get the accuracy similar to the low resolution structures. And if you get these structures, you can get the atomic coordinates and this can be used for docking of small ligands, or you can see the interactions, because you will get high quality structures. And if the sequence identity is less, for example 30 to 60%, in this case you can get the accuracy similar to the molecular replacement in crystallography. How the molecular replacement in crystallography works? Because there is based on homology, if there are similar electron density maps, they can see that they may have similar structures. Like that level you can get the structures if the sequence identity is about 30 to 60%. So, if you want to do scientific mutagen studies, these models you can use. If the sequence identity is less, you can get a model, it is very crude model. In this case it is not good for the very high resolution, like high resolution structures, and we cannot get all the interactions, but we can try to use it for assigning the fold, or you can see the contact between residues if you consider C alpha atoms, right, because the models are very crude models, we do not get the well defined structures, but you can use it to understand what could be the probable fold or which residues are possible to interact with each other and so on. Right, now the question is, what is the sequence identity we require for homology modeling, right, where you require 30% or 50% and 60% and so on, right. What is the average percent accuracy, the identity required for homology modeling? Right, that you can see the number of aligned residues, right. If we have 150 residues which are aligned, and if the identity is more than 50%, like see here, this you can say as a safe zone. If it is less number of aligned residues, even the sequence identity is very high and the aligned residues are high. So, because we will get same sequence, different structures. So, in this case the homology modeling will not work, because we will adopt different types of structures. So, if we have the 100 residues, then we need at least 60% sequence identity, depending upon the number of aligned residues and the sequence identity, right, we can select the cutoff identity for homology modeling. Here I mentioned that 150 residues with 50% is a safe zone. In this case you can use for homology modeling, and you will get reliably good structures, right, using the structure prediction algorithms. So, when you make the homology modeling, right, what are the various steps one has to consider for homology modeling, right. The first one is we need to identify a template, your query sequence, right. You have to identify a template, because your model you build based on the template. So, it is very important to identify a proper template, right. So, because your structure finally depends on the selection of templates. So, it is very important to take the proper template, right, and then you need to do the alignment. How to make the alignment? Structure. Right, you can do the initial sequence alignment using the BLAST, right, and if you look into the alignments, so you check any specific residues which are conserved or any residues which are having important functions, these residues should be properly aligned, right. If these residues are not properly aligned, finally you end up with the structure, right, which is different from what you require. So, you need to do the proper alignment and check the alignment whether the important residues, right, with the experimental information are properly aligned or not. Once you do that, you can make the corrections and finally make sure that your alignment is correct, right, with respect to the experimental information available for the particular protein. How to get the experimental information for a specific proteins? Either use the literature, or you can also get from the Unipro database and so on. Once we get the alignment correction, then you go with the backbone generation, because we get the template, your template is ready. Then for your target you can just copy the backbone, because the backbone is the same, right, the target and the template, and use this as your to build the model. Then if you see there are proper alignment of residues, the residues are the same, then you can copy the side chains, right, and other cases you can replace these amino acid residues. Then you do the loop modeling, right, for there are several loops, then you have to get the modeling the loops, right, and then you do the side chain modeling. The side chains can take several conformations, right, you can use the available conformations to model the side chain, and once the model is built, then you need to optimize. Whether you check the model is energetically favorable, right, you can optimize the model. So, finally, if you are happy with the model, then you validate whether this model is valid based on the energy or the bond length, bond angle, torsion angle and so on.